A very good afternoon. My name is Jessica. I'm a Year 2 postgraduate student from the University of Nottingham Malaysia, and I'm here today to share with you about my project, which has recently been awarded with the MAKNA Cancer Research Award. So without further ado, I'm going to begin first by giving you a bit of background about my research topic focus, which is ovarian cancer. So ovarian cancer, as you can see, is the cancer of the ovaries, and as we know, it's the female reproductive organ. And the thing about ovarian cancer is that it progresses very fast. So the symptoms of ovarian cancer are often overlooked. They are very vague, so women who experience the symptoms think that it's nothing, it's something minor. So, for example, bloating, abdominal pain, change in bowel habits. The current diagnostic methods include a blood test called serum CA125. We can also do ultrasound, abdominal CT scan, and biopsy if we want to diagnose it. In Malaysia, it is the fourth most common cancer, and according to recent data published last year, 56.3 of cases, ovarian cancer cases in Malaysia, were detected at stages 3 and 4, which is the late stage. Okay, so by this time, the cancer has already metastasized to our brain, to the lung, and also the liver. So it's very aggressive. So a lot of cancer patients don't survive, or their survival rate after 5 years is very low. So here are some of the disadvantages of current methods. Basically, the most common and cheapest, I would say, is the serum CA125 analysis. So we can see that its sensitivity is only 88.6%. This means that it doesn't detect very low, it cannot detect really early on. And for specificity being 72%, this indicates that it's not specific to just ovarian cancer. It will also be elevated in the blood if a woman has endometriosis, uterine fibroids. So therefore, we get a very high false positive rate with this method. The other methods like ultrasound and abdominal CT require special equipment or specially trained personnel to use this equipment and to interpret the results. Okay, ultrasound does also suffer from a high false positive rate, and abdominal CT, as you know, is the CT scan. It's not widely available and it's quite expensive to do. Okay, so I'm now going to talk about another potential biomarker for the cancer, and it's called miRNA-665. So microRNAs, or miRNA for short, they are basically non-coding RNAs. In the case of miRNA-665, it's a very short nucleic acid. It's only 20 nucleotides long, right? And one of 10 miRNAs, okay, were reported to provide 99% sensitivity and 100% specificity for distinguishing the ovarian cancer patients from non-cancer patients. So this is one of the 10, all right? So what miRNA-665 has been found to do is that it promotes proliferation or multiplication of the ovarian cancer cells, all right? Now these microRNAs, they can actually circulate in the bloodstream in a stable form, which means that if we collect a patient's blood serum and we analyze it using microarrays or RTPCR, we can detect this microRNA in the patient's blood serum. So the thing about the current methods like microarrays, you can see that it takes quite long, approximately 24 hours. RT-PCR is, you can say, it's the standard method. It's not too long. It's approximately 4 hours, and this is used for COVID-19 screening as well. Yeah. So I'll be talking a bit more about reverse transcription polymerase chain reaction. Okay, so RTPCR for short, it requires special equipment and skilled personnel. Okay, so first and foremost, we need a thermocycler. We have our microRNA, which needs to be converted into cDNA, and then this cDNA will be amplified with the help of this machine here. This entire process requires enzymes, something called thermal cycling, which is where you vary temperature, and it has to be very specific for how long, right? And you have to accurately design your primers for this to work as well. After you have amplified, then with traditional conventional PCR, we visualize with gel electrophoresis. It's quite time consuming. You need to be trained to do it. It needs DNA staining dyes and a rather expensive piece of equipment called a gel imager. Alternatively, qPCR, which is a more advanced PCR, enables real-time quantification, so we don't need to do gel electrophoresis. However, it uses a special primer which is labeled with fluorescent markers for fluorescent dye. So these primers increase the cost of qPCR quite considerably. Yeah. So basically, my project aims to achieve miRNA amplification without the use of enzymes, and this will be done by hybridization chain reaction. Okay. So in 2004, two scientists named Dirks and Pierce, they came up with this DNA circuit system, and they termed it hybridization chain reaction. So essentially what it is you have DNA probes which are designed such that they form a hairpin. So it looks like a hairpin. Once it's in the presence of a nucleic acid target, it can be a DNA, it can be a microRNA. The target will cause the first hairpin to open up because it can bind to it. When it binds to it, it pushes the I've used colors to represent this portion here. It pushes the hairpin open. When that hairpin opens, it exposes a region that now H2 can bind to. So in other words, you get a chain reaction because now they can just self-assemble on their own, and it repeats until you get a long double-stranded DNA. And this happens without enzymes. The second stage of my project is basically using gold nanoparticles to achieve colorimetric detection. Okay, so gold nanoparticles, or AUNPs for short, produce optical signals which we can see with the naked eye. Okay, so for example, when the gold nanoparticles are dispersed in solution and they are far apart, they appear red in color. However, if something causes the nanoparticles to aggregate, they move closer together, they become blue in color. So the reason for this is because the interaction of DNA with the gold nanoparticles, which is what I'm going to exploit in my project. Basically, in a hairpin form, the hairpins might be able to stabilize the gold nanoparticles I'm using. And after HCR, in which the hairpins become a long double-stranded DNA, they don't interact in the same way with the nanoparticles, and it is the hope that it will aggregate and it turn blue. So research has been done primarily with a certain kind of gold nanoparticles. However, I'm going to be using cysteamine-capped gold nanoparticles, alright, because they have been successfully used to detect asthma drug called clenbuterol, viral RNA, plasmid DNA. However, they've not been used to detect microRNA yet. Okay. So my project goal is to achieve enzyme-free detection of microRNA 665 with the cis and AUNPs. Okay, so basically this is what my proposed assay would look like. First, I would have my DNA probes, 3 different kinds, and they would all be partially complementary to each other. Alright, I intend to incubate with the patient blood serum or serum spiked with the target, my microRNA 665, and after 30 minutes, the hairpin probes would actually assemble to form branch DNA. So because of the difference in the structure, okay, the cis AUNPs that we then incubated with could either remain red or it could turn blue. So one hypothesis is that when the miRNA 665 is absent, it would remain red, but if it is present, it would turn blue. So this way we can see with the naked eye, and the results will be fast as well. So the advantages of the HCR cis AUNPs hybrid assay is that number one, it's instrument free. We don't need special expertise or in-depth training. We can get fast results with it. It's enzyme free. So being enzyme free means that it's more robust. We don't have to worry about storage temperature so much. It's not so sensitive to the environment, and also brings down the cost. Finally, it works at constant room temperature, so we don't need equipment like thermocycler. Yep. So all in all, this makes the assay suitable for point of care testing in low resource settings. So let's say in rural areas where the clinic doesn't have access to expensive laboratory equipment, or even in urban areas where the general physician's clinic has limited resources, but something simple like this would be able to be used anywhere. So this achieves our ultimate goal, which is to facilitate early detection of ovarian cancer, because as mentioned in the first slide, ovarian cancer is detected late, and this contributes to a much lower survival rate. So if we can detect ovarian cancer early on, and it's made available to more women, then they can get treatment earlier if they are diagnosed. And if they get treatment earlier, then they might actually survive and not have the cancer reoccur in their ovaries. So yeah, these are the references, and that's all for my presentation. Thank you.